MicroRNAs are a class of non-coding RNAs that play roles in regulating gene expression. In this episode, we'll discuss discovery of microRNAs, their function, transcription, mechanism of action, and their applications. This is Alia Aracha, and you're watching Denable. Stay tuned. Discovery of microRNAs. MicroRNAs were discovered in 1993. Before that time, scientists have never imagined that RNA could have the ability to regulate DNA. So the function of microRNAs is to regulate gene expression, whether by inhibition or promotion. Transcription of microRNAs. Like any gene, transcription of microRNA requires transcriptional factors and RNA polymerase II. So the microRNA can be transcribed from the DNA into the pre-microRNA. The pre-microRNA is then processed by an RNase called drosha, then exported to the cytoplasm by a protein called exportin 5. In the cytoplasm, the pre-microRNA is processed by an RNase called dicer into microRNA duplex. The microRNA is usually combined with the risk protein, which is necessary for the microRNA mechanism of action. The microRNA's mechanism of action is to either bind to DNA, which is uncommon pathway, or to bind to mRNA, which is a traditional pathway. By binding to DNA, microRNAs can activate or inhibit the transcription of a gene. Binding to mRNA causes the inhibition of translation of that mRNA. First, let's discuss the traditional pathway of the microRNAs. This is the risk protein guided by the microRNA to the complementary site on the mRNA 3' untranslated region. The risk protein can either cause degradation of mRNA or just inhibit its translation. At both cases, we have reduction in protein translation. This is the microRNA regulation at a post-transcriptional level. At the transcriptional level, microRNAs can either activate or inhibit gene expression. So the second mechanism of action of microRNAs is to activate gene expression. MicroRNA can be directed to the promoter site by binding to the non-coding promoter-associated RNA. The promoter is a site located upstream and controls gene expression. In this case, microRNA is attached to the activator complex, which enhances the gene expression. The third mechanism of action is to inhibit the gene expression at a transcriptional level. In this case, the microRNA makes a complex with inhibitory proteins. This complex is guided to the promoter site either indirectly via binding to the non-coding promoter-associated RNA or directly via binding to the promoter. The inhibitory complex causes the inhibition of gene expression. Applications of microRNAs. Examining the microRNAs can help in early diagnosis of some diseases such as cancer. As the main function of microRNAs is to regulate genes, microRNAs can be used in gene therapy for treatment of many diseases. These are the references, and thanks for watching.